Hello everyone, I'm Pinar Demetci and I'm a second-year PhD student in the Computational Biology program at Brown University. I'll be presenting a new unsupervised learning algorithm that uses Gromov-Wasserstein optimal transport to align single-cell multi-omics data. My co-first author, Rebecca Santarola, is also delivering a short presentation on this. Let's define the problem we're addressing. Single-cell sequencing has allowed us to study the genomic heterogeneity in cell populations. With the availability of various sequencing assays that capture different aspects of the genome, an important challenge in the field now is to integrate these disparate types of data. Availability of co-assays on single cells, where multiple sequencing technologies can be applied on the same cell, is still very limited, and for some combination of sequencing methods, it's not possible to perform co-assays, since they may require access to the same part of the genome and interfere with one another. This means single-cell sequencing data usually comes from different batches of cell cultures, where cell-to-cell correspondences are unknown. And since these sequencing methods profile different aspects of the genome, we often don't have feature-wise correspondences either. As a result, in order to get a unified view of the genome, we need unsupervised algorithms that align these data in some shared space without requiring any correspondence information. Here, we present a new algorithm called SCOT, which stands for Single Cell Alignment Using Optimal Transport. Through benchmarking experiments on simulated and real-world datasets, we show that SCOT successfully aligns different sequencing data in a computationally efficient way and reduces the hyperparameter search space in comparison to the previous unsupervised methods. SCOT relies on a special type of optimal transport algorithm called Gromov-Wasserstein optimal transport. Optimal transport aims to move data points between different distributions of data in the most cost-efficient way possible. What's special about Gromov-Wasserstein optimal transport is that, unlike traditional methods which compare data points directly, it compares intra-domain distances to find the optimal transport plan and defines the cost of transport over these distance measures. This allows us to preserve some local geometry during alignments and align domains of different dimensionality. Our algorithm operates in 3 steps. Given 2 domains of different sequencing data, we first extract intra-domain distances by constructing k-nearest neighbor graphs over the samples in each domain. K is a hyperparameter here, but our algorithm is robust to the choice of k. We represent these graphs as adjacency matrices, which store sample-to-sample distances. Then, given these matrices, we formulate our optimal transport problem as shown here. The goal in this optimization statement is to find the coupling matrix gamma, which stores sample-wise correspondence probabilities between the 2 domains. It tells us how likely it is for a sample from one domain to be aligned with the sample from another domain. Ideal gamma matrix is the one that minimizes this cost defined over the Euclidean distances between the intra-domain distance matrices, so that the overall geometry of one domain will look like the other after transport, while still preserving relative pairwise distances within a domain. We also include an entropic regularization term over gamma here for computational efficiency and also for obtaining better empirical results. Epsilon here is also another hyperparameter. We have 2 hyperparameters in the algorithm. We solve this optimization problem iteratively with a matrix scaling algorithm called Singhorn-Knopp iterations, which makes sure that probabilities in each column and also each row of the gamma matrix add up to 1. While this coupling matrix might be sufficient depending on the application of the alignment, we transport samples from one domain to the other using barycentric projection. This projects data points from one domain based on the weighted average of the data points in the other domain, scaled by the correspondence probabilities. We applied our method to simulated datasets and real-world co-assays where we had some ground truth on cell-to-cell correspondences to analyze the quality of alignment. We benchmarked our performance against 2 unsupervised algorithms in the field, MMDMA and UnionCom. UnionCom is also being presented at ISMB. We performed hyperparameter tuning for all the methods and compared the alignment accuracies based on the average fraction of samples that are aligned closer to a sample than that sample's true match. So lower values of this metric are better. Here we have 3 simulated datasets: bifurcation, Swiss roll, and circular frustum. In the first two columns, you can see the projections of the original domains. We visualize the alignments performed by Scott on the third column. On the fourth column, we show that Scott achieves the best alignment results for all simulation datasets and completes the alignments under significantly less time. Moving on to the real-world datasets, we use data from SCGEM sequencing and SNARE-seq, both of which are co-assays. SCGEM simultaneously profiles gene expression and DNA methylation on the same cell, while SNARE-seq sequences gene expression and chromatin accessibility. Again, on the third and fourth column, you can see the alignment results. For SCGEM, all three methods perform similarly, but for SNARE-seq, we see that MMDMA yields the best alignment, followed by Scott. In terms of computational time, Scott is still the fastest. Overall, we show that Scott yields alignments comparable to the other state-of-the-art unsupervised alignment algorithms, but in significantly reduced time, which is desirable when working with high-dimensional datasets. Moreover, while UNION-COM and MMDMA require tuning of 4 hyperparameters, Scott only requires 2 and is robust to the choice of 1. This is an advantage because tuning hyperparameters can be challenging in an unsupervised setting. Currently, we're looking into extending the application of Scott to more than 2 domains at once, and comparing under which circumstances Scott and MMDMA may be preferable over one another. We're also exploring different projection strategies as well as different uses of the coupling matrices. Our preprint is owned by Archive if you'd like to look at it, and thanks for listening.